Well, hello, everyone. Welcome, members, and happy Members Month. Yay, woo. We are super excited for you all to be here. We have folks joining us virtually and in person today. We are also so happy to have our speaker, Elora López-Nandam, who is an Academy researcher. She will be sharing with us some closer looks into the Academy's Coral Spawning Lab and how the genomic data is being used to support Hope For Reefs initiative. We will have a Q&A at the end, and we'll have plenty of time to answer questions. Without further ado, please join me in welcoming Elora.
Thank you so much, Jess, and thanks to all of you for being here. This is the first time that I'm giving a talk in person in over 2 years. Thank you. I really wasn't looking for the applause, but I appreciate it. So I'm so excited to be here with you today, and that we're talking about the Hope for Reefs initiative, as well as the Coral Spawning Lab here at California Academy of Sciences. Today, I'm going to tell you about what we're doing in the lab, but before we get to that, I want to make sure that we're all on the same page. And first asking this question, what is a coral reef? So what you're looking at here is an image taken from space of an enormous ecosystem that's made up of millions upon millions of coral animals. When we zoom in and go a little bit under the ocean surface, we start to see that this is an ecosystem made up of the corals themselves. So these are different coral colonies of different species here on the bottom. And you can see that these corals are made up of what's called calcium carbonate skeletons. So what I'm holding here, and hopefully for those of you in person, you can come down and check it out after the talk, is a coral skeleton. So this is made up of calcium carbonate. This animal no longer is alive, but when it was alive, this skeleton supported a very thin layer of living tissue over it. And so it's these structures that actually create these incredible three-dimensional reef structures that can be seen from space. And because they create such complex and interesting structures, these are really incredible ecosystems for supporting biodiverse fish and other animal life. But what is a coral? So I gave it away a moment ago. These are actually animals. So contrary to what you may believe and contrary to what they may look like, they're not rocks and they're not plants. These are actually animals that are part of the group Cnidaria. So cnidarians are a really ancient animal group that includes sea anemones, jellyfish, soft corals, and stony corals. Today, we're going to be focusing on stony corals. Again, the reason that they're called stony is because of these calcium carbonate skeletons. So if we start by looking at a coral colony, this is a colony that is about 2 meters across. You can see me measuring it during my dissertation work in American Samoa. The reason that we call this a colony becomes a little bit more obvious as we start to zoom in. So a colony is made up of individual coral polyps. So you can see I'm circling a coral polyp here, and each polyp in and of itself is a multicellular animal. So each polyp has its own tentacles, its own mouth, its own gut. And all of these polyps are living together in this calcium carbonate skeleton. I often imagine it as people living in an apartment building together, except you and all of your apartment mates are clones of each other. So the way that a coral colony grows is that an individual polyp divides into two and keeps dividing and dividing. So the way that a colony grows is actually by asexual reproduction. If we start to zoom in a little bit more on each of these polyps, and then even more to a single individual polyp, you'll notice that the polyp tissue itself is incredibly translucent. It's almost entirely colorless. The only pigmented cells that you see here, these green cells, are actually not the animal cells, they're algae. So we'll talk about this more a little bit later, but the coral-algal symbiosis is incredibly important. The algae that lives inside the coral's tissue take energy from the sun and convert it into food for the coral animal through the process of photosynthesis. And we'll talk a little bit more about why that's really important today. But first, let's think about why we should care about coral reefs. So we first hit on this one a little bit already, biodiversity. So you may notice in this figure here, this is a map of the world and where we see coral reefs. And on the second plot here, you'll see where coral diversity is the highest. So when we get to these hotter, redder colors is where there's more species in a given area. And you'll notice that as you increase in coral species diversity, you also increase in the number of fish species that are in that area, the number of snail species, and the number of lobster species. And so as we have more diverse reefs, we have more diverse ecosystems of other species as well. And this is really important for fisheries. So fisheries can span anywhere from small scale subsistence fishing, and millions upon millions of people around the world today still rely on subsistence fishing in order to really catch what they need to eat for the day, every day on coral reefs, all the way up to much larger industrial fishing vessels. And even if these fishing vessels are not fishing on the reef itself, even though coral reefs only cover just 1% of the ocean surface, they support 25% of fish species that live in the ocean at some point in their lives. So even if they're not being caught on a coral reef, many of these animals that are being caught in industrial fisheries were born or spent a lot of their juvenile phases on a coral reef. Another reason these reefs are incredibly important is coastal protection. And this is something that even the US military is really starting to want to invest in. So there recently was a program that was launched to try to mitigate coastal flooding, erosion, and storm damage by protecting coral reefs. So if you've ever been on a beach where there was a reef, you may have noticed that the waves were not breaking on the beach itself, they were breaking on the reef. And that means that as you have healthy coral reef ecosystems, the wave energy is absorbed by the reef itself, and then there's way less wave energy by the time the wave gets to the shore. So as we have eroding coral reef ecosystems, we run the risk of more flooding, more erosion, and more storm damage. Another reason these reefs are incredibly important is because they support recreation and tourism. Of course, everyone wants to travel to reefs, and there's many economies around the world that have a huge component of the economy based on recreation and tourism. We also can't forget the cultural significance of reefs. So this is a line from the Kumulipo, or the Hawaiian origin chant, which says, "Born was the coral polyp, born was the coral, came forth." This is one of the first lines of the chant, and it really emphasizes the importance of the coral as the beginning of the creation of the world. And so there's incredibly diverse societies all around the world that emphasize coral reefs as being incredibly culturally significant. And then finally, this is often a little bit overlooked, but corals are really a model for genome integrity research. So this animal that you're looking at here is called Big Mama. She's been estimated to be 535 years old. And clearly, as you can see, she's very large as well. So animals like Big Mama don't show aging in the way that we usually think of human aging. And corals don't generally get cancers. And so there's a lot of information in the genomes of these animals that we might be able to uncover how they actually do these things. So hopefully I've started to convince you that these reefs are important, which then begs the question, what's happening to coral reefs right now? So there's lots of different local stressors, coastal development, overfishing, runoff from agriculture. But today I really want us to think more about the global pressures. So ocean acidification and coral bleaching from global warming. So briefly, ocean acidification. As our atmosphere is absorbing more carbon dioxide, or CO2, a lot of that carbon dioxide is getting absorbed by the ocean. And this becomes carbonic acid in the water. This is a huge problem because carbonic acid can actually dissolve the skeletons, the calcium carbonate skeletons, not just of corals, but of snails, urchins, mussels, all sorts of other marine animals that have these calcium carbonate skeletons. And then finally, global warming and coral bleaching. And you may, even in the last week, have heard that there's bleaching going on yet again in the Great Barrier Reef today. So what is coral bleaching? Well, earlier we talked about the incredible symbiosis between the coral animal and its algae. But for reasons that we don't fully understand yet, and scientists are actively trying to figure out the mechanisms, when ocean temperatures rise, corals start to expel their algae. And so the reason that this phenomenon is called coral bleaching is because the animals literally turn white because they've lost those pigmented algal cells. So what does that actually look like for a coral reef? Well, this is a healthy reef and what it looked like in December 2014 in American Samoa. So these are a whole bunch of very healthy yellow-brown pigmented staghorn corals. This is what they look like 2 months later due to coral bleaching. So they've lost all of their algal cells. At this point, the animals are still alive, but they're in effect starving to death. Because as we mentioned before, the algae are the ones that are producing a lot of the food, up to 80% of the food for the coral. And so if the animals aren't able to get their algae back in time, then you end up with a dead reef within a matter of months. And you'll notice that in this photo, the reef structure is still there, so the skeletons are still intact. And so fish and other animals still can live on a dead reef for a while. But after a certain point, when these animals are no longer alive and able to repair themselves, the reef structure starts to degrade as well. And so that eliminates a lot of habitat for a lot of other animals. So how are scientists trying to protect corals? Well, we know for sure that the most important factor in how corals are going to fare in the next century is how much we decrease greenhouse gas emissions and how soon. But even if emissions were to drop tomorrow, ocean temperatures will still be on the rise. And so restoration efforts are on the way to try to bolster coral populations and hopefully help them to adapt to new conditions. And there's a lot of different ways that different people are going about this. One of them is restoration via fragmentation. So if any of you have ever worked with cacti or any types of succulents, you might know that you're able to cut off a branch or break off a piece and plant it somewhere else. Well, you can do the same thing with a coral. And so these coral gardens are popping up in places all around the world where people are breaking corals into different pieces and replanting them onto degraded reefs. So this is great in the short term, but the problem then is that all of these new outplants are very, very genetically similar to each other. In effect, you're creating gardens of clones. And so if a stressor like warmer temperatures or disease or something else crops up that that particular genotype can't deal with, you might eliminate an entire clonal reef very quickly. So here at Cal Academy, what we're really excited about is starting to work more with coral sexual reproduction. So the species that I'm going to be talking about today is an animal that's capable not just of asexual reproduction, but sexual reproduction as well. And we'll get way more into the details of what that looks like, but very generally, we have an adult coral that releases eggs and sperm. So both eggs and sperm are released by a single animal in our species. And those eggs and sperm from different colonies then find each other in the water column. So they fertilize each other, and that's when you get an initial embryo. Those embryos then develop into little swimming larvae. So we go fertilize to embryo to swimming larvae. And the larvae swim around in the water column for a few days to a few weeks. And then hopefully they find a nice place to settle, they settle down, and they start growing as a coral colony. So the question then is, how can captive breeding and studying this sexual reproduction process actually provide hope for reefs? And that's where Cal Academy's Coral Spawning Lab comes in. So the Albright Coral Spawning Lab was started by my boss, Rebecca Albright, in 2017 here at Cal Academy. You can see our system on the left. So the amazing thing about this lab is that we're able to control all sorts of different environmental factors. So we have all of the lighting system synced up to the lighting in the location where the corals came from. So all of the solar and lunar and day-night cycles are all the way that we program them to be, which is really important because we'll get into this in a little bit, but spawning is entirely based on lunar and day and night cycles. So first, I want to tell you about some of the initial successes in 2019 and 2020 in coral spawning in Cal Academy, and this particular species, Acropora hyacinthus, or the tabletop coral. So we have a really nice video that Rich Ross created based on all of the work that was done in 2019. So here we are in San Francisco, the coral spawning lab being built behind the scenes in the aquarium. And then the animals in 2019 and 2020 were sourced from the island nation of Palau. So a team from Cal Academy went out to Palau and see which colonies look like they're going to spawn within the next few months. So they're looking for animals that already have eggs and sperm developing. Once they identify them, they take off just a piece of a colony, not the entire colony, and they pack it for shipment back to San Francisco. Once the animals arrive in San Francisco, there's a lot of processing that has to be done, and then they get settled down into the tanks in the spawning lab. And then just a few days after the full moon, we start to see this. So what you're looking at here are bundles of eggs and sperm being released into the water column. So as this is happening, our scientists are collecting the eggs and sperm and starting to create combinations in order to produce embryos. So what you're looking at here is a huge vat of eggs and sperm, and the embryos are just beginning to develop. It's very exciting. So here you see embryonic development. So the animals are just starting to spin. And here you see coral larvae. You'll see that they're swimming on this penny here, so they're very small. And although they're small, they're actually capable of swimming around and actually sensing what's around them to find a place to start settling and metamorphosing. After a few weeks, we start introducing algae, hoping that they'll uptake them. And in 2019, this was a huge success. So we ended up with hundreds of thousands of larvae and lots of settlers. So we were able to collect material to create a full new genome assembly for this species, Acropora hyacinthus. It's actually now the best assembled coral genome in the world. But I wanted you to remember for just a moment what you just saw about a minute ago, which were those first initial little polyps that just had settled. So those little white polyps settled on their discs. So that was in May of 2019. Do you want to know what they look like 2 years later? This is an incredible amount of growth in 2 years. So in 2 years, we have juvenile corals. These animals are now, the 2019 ones are now 3 years old, and they're large enough that we're actually going to be checking them tomorrow to see if they might start reproducing next month. So this was incredibly exciting and a huge success. And really the first time that this system had been developed to be done in the US. We were the second in the world. The Horniman Museum in London was the first to really develop this ex situ spawning, but Rebecca's team was really the first in the US. So I joined the team a year ago this month, actually, and my background was in coral genetics. And so I was really excited about the husbandry that was being done in the lab. And what I'm hoping to do is to combine that husbandry with genomics to inform coral conservation. So last year, we sequenced the genomes of all of the surviving offspring that had been born in 2019 and 2020. We also sequenced the genomes of their potential parents. So up until this point, we didn't know how any of the animals in our system were related to each other, which ones were siblings, which ones were parent and offspring, nothing like that. So we sequenced the genomes of 3 animals that had spawned in 2019, as well as their 23 babies that had survived to be 2 years old at that point, as well as 13 spawners in 2020 and their 9 babies that had survived to be 1 year old by that time. And my first objectives here were to create pedigrees or family trees to figure out the relationships between all of these animals. And then next, I was interested in starting to figure out if there's signals of selection induced by the lab environment. And by that, I mean, we of course know that having animals reproduce in the lab is very different from having them reproduce in the wild. And undergoing development and becoming a juvenile coral under lab conditions are incredibly different from the wild. And so we're interested in figuring out if there are actually genetic variants that are more suited to surviving the lab environment than in the wild. But first, let's look at some of these pedigrees. So these are the relationships of the animals from 2019. So the first thing you'll notice is this enormous family tree on top. So these 2 parents, 56 and 60, were the parents of 22 of our 23 surviving babies that survived to be 2 years old. Whereas these 2 parents only had 1 surviving offspring that survived to be 2 years old. So there's already, just by looking at this information, a lot of questions here. So why were 56 and 60 so good at producing offspring compared to the other 2 parents? One possibility is that they each produced way more eggs and sperm than the other 2, and so it's just a probability issue. But another possibility is that these numbers 56 and 60 had genetic variants that were potentially already better adapted to having their offspring survive in the lab environment. And so this is a question that we're actively pursuing in the lab. So if we look at the 2020 animals, we start to see some similar patterns again. So first of all, we had a coral love triangle happening here. So you see that coral number 74 and 80 had 4 full offspring together. So these animals are all full siblings of each other. But these 4 each have 2 half siblings as well, because 74 and 80 also had an offspring with 71. You'll notice over here, 58 and 63 had 1 surviving offspring. But a really interesting thing to note here is that the majority of our parent corals, or would-be parent corals, they spawned, but didn't have any offspring survive to be 1 year old. And so again, our question here is why? And this is something that we're actively trying to understand. So part of that involves identifying gene variants that are under selection, which is something that, again, is really a work in progress, but I want to acknowledge my collaborator and friend, Cheyenne Payne, who's a PhD student in biology at Stanford, who's actively helping us try to untangle this question. But now I want us to move into the December 2021 spawn. So this was our spawning event that just happened a few months ago, and it was really exciting for me because it was my first time leading and really being actively involved in every step of the process. So I quickly want to acknowledge, again, Rebecca Albright, who made this possible, but also Lisa Larkin, Stephen Young, and Charles Delby, who are amazing Steinhardt Aquarium staff. So they're the ones who are in charge of keeping our animals alive and healthy and do an incredible job every day. And then also Shale Matsuda, who is a postdoctoral researcher at Shed Aquarium in Chicago. So Shale came out to help me really be in the trenches every day during the spawning process. So let's talk about what that process actually looked like. So our corals first arrived here at Cal Academy in October of 2021. So join us behind the scenes here as these animals are just getting loaded off the truck after a very long transit from Cairns, Australia to San Francisco. And once these animals actually arrived here at the Academy, there's a whole bunch of things that had to happen on that very first night. So first we have a process of unboxing the animals. So the way that these animals were packaged for shipment is they're first put into truly just a plastic bag of seawater. Those plastic bags of seawater are then put into styrofoam boxes, and the styrofoam boxes are put into cardboard boxes. So it's really not that high tech, but amazingly it works. You can see our coral biologist, Lisa Larkin here, inspecting our animals upon arrival. And then for every animal, as we unpacked it, we took off a branch and stored it for later genetic analysis. And we put a plastic tag on each of our corals so that we could track it and keep track of it throughout the duration of its life here at Cal Academy. So here is Shale putting our samples into liquid nitrogen. That's how we preserve them for DNA extraction later. And then we had Carissa Mendoza, who's part of our amazing water quality team, checking the water quality of each of the bags that the corals arrived in. And then finally, after some dips to make sure that all of the animals were healthy, they ended up settling into our system. And so I was very happy that by the end of October, we had all of these very happy and healthy looking animals. So again, these are Acropora millipora. They're called the dragon scale coral, and they were collected in the northern Great Barrier Reef, so from 3 different reef sites. So part of the thing that we wanted to understand is how corals from other reefs were compatible or not compatible to produce offspring with each other as well. So you'll notice that we have little fish and sometimes little snails or urchins, sometimes even little crabs living in the system to keep the animals healthy and clean as well. So this is the end of October. The corals are settling into the system, and we knew that we were going to start expecting to see some spawning potentially in November. The reason for that is that these animals only spawn once a year. They spawn once a year, just a few days after the full moon in November. So we had a few weeks to wait, and there were a lot of preparations to be done in the meantime. So we have Lisa, as well as Ryan Schaefer, another amazing aquarium biologist here, setting up larval cones where the larvae would live. So these cones have very highly filtered water going into them, so that the water is really clean as the larvae are first developing. They also have just a gentle amount of oxygen being pumped into them every day. So there's a lot of preparations happening on that side. And then Shale and I took over a conference room just a few feet away from the lab and turned our conference room really into a mad scientist lab as well. All right, so then it was November 2021. We knew that the full moon was going to be on November 19th. And so that is the day that we first started checking for coral spawning. And the way that you check for coral spawning is the entire room is dark. The reason for that is that we know these corals spawn just a few hours after sunset. So we do something a little sneaky and make sunset in our system happen at 11:30 a.m. That way we're working in the afternoon and not in the middle of the night. So from about 11:30 a.m. to about 3 p.m. every day, we go in every 15 minutes in the dark wearing red headlamps, and basically in the dark with just red light, start shining our lights on the animals. And what we're looking for is a process called setting. And setting is, if you're looking at the coral polyp, so my fingers are little tentacles here, setting is when the gamete bundle starts to actually be visible, and you can actually see the little tentacles holding its gamete bundle. And so this, when you actually see that happening, you know that spawning is going to happen in the next 20 minutes to about 2 hours. So this is what we're looking for. So we start November 19th, November 20th, no setting, go through November 25th, no setting. November 25th, by the way, was Thanksgiving. So this is us enjoying Thanksgiving in the lab. We got to the end of the November, still no setting. We were starting to get a little bit worried. And then finally, on December 2nd, I was in the lab by myself. I had been staring at these corals for weeks. So my eyes, I felt like sometimes were playing tricks on me. And I was looking at this coral and I was like, oh, this might be it. I'm not totally sure, but I started to text Shale to tell him, I think this might be happening. And just as I looked at my phone, I hadn't sent it yet. I looked back at the coral and I was like, oh no, this is it. So that's when you make the call and say, okay, everyone, let's go. So we had 3 corals spawn that day, which was great. But even better was the next day when 9 colonies spawned. And this is really exciting because you really want your corals to all spawn on the same day so that you can have as much genetic diversity as possible, because otherwise those eggs and sperm just aren't going to meet each other. So we had no spawning on the 4th, and then on the 5th, we had our last day with just 1 animal. So at first we were a little disappointed that that 1 animal had waited so long, but it turned out to be okay because it became a movie star. This is in real time, by the way. Sometimes people think that we have messed with the timing, but this is actually the rate at which these gamete bundles get released. So the reason that it actually turned out to be really nice that we were able to get footage of just 1 animal that we weren't going to use for the science is because this is what it looked like when we had all of our animals spawning. So again, we're in the dark, we're using just red lights. And our corals are wearing these really funny little coral nets. So the reason for these coral nets, these are what we put on them as the corals look like they were about to start spawning. So we have a colony sitting under its net. All of the gamete bundles are buoyant, so they float up really nicely into the cone, and they drift right up into our falcon tube here. So you can see all of the gametes collecting into the tube. And then once we're ready to actually take the tube off, so once the corals have all stopped spawning, all you have to do is unscrew the tube still under the water, put a cap under it, and then we're ready to go. So this was actually a really ingenious invention that Shale helped me create here. So once we have all of those genotypes, we can then start combining them into one gigantic fertilization pool. So this is us feeling very pleased with ourselves. And this is what all of the eggs and sperm look like when they're all pooled together, all 9 colonies. So what you're looking at here, the colored dots that you actually see are all eggs. So there's lots and lots of eggs in here, but for every 1 egg, there are millions of sperm. So for every gamete bundle that these colonies produce, there's about 5 to 10 eggs and 10 million sperm. So this is an incredibly dense amount of gametic material here. So they stay in the bucket for about an hour, and then they get transferred to the larval cones that you saw the setup for earlier. So this is what the larval cones look like a few days after spawning. You can see kind of through the side, there's a bunch of little larvae. We'll open one of the cones up in a moment and you'll actually be able to see a little bit better. And I will say this was so many more larvae than I was expecting. We thought that we would be really happy if we filled up 1 or 2 cones, and we ended up filling all 5. So you can see how dense they are in there as well. They're really actually kind of starting to form clumps together. So these are millions upon millions of larvae. So at each stage, I wanted to actually take a sample of our embryos and larvae. So this is what all of our embryos look like when they were 4.5 hours old on December 3rd. So this is pretty incredible because you can actually see, like, this is a 2-cell embryo in the stage of division. This is a 4-cell embryo. You know, these are incredibly early stages of development. And so at this stage, not only did we take images of them, but we took a small subset of them and saved them for later genetic analysis. Because one of the things that I'm really interested in is seeing how genetic variation changes in a population over the course of just the first few days of development, because that might start to tell us which genes are actually the most important in allowing a coral embryo to survive into adulthood. So this is what they look like at 17.5 hours old. They're starting to become this kind of flat chip-like structure. This is called the prawn chip stage of development. And then by 24 hours, they're starting to become a little bit more three-dimensional. This is the donut stage. And then at 40 hours old, we're starting to see a little bit of spinning, and that's because these animals now have cilia. And then by 52 hours old, these guys are zooming. So at this point, they're still very circular. Over the next few days of development, they become a little bit more elongated, and I'll show you what an elongated larva looks like in a little bit. But this is really interesting information, and the samples that were taken in these couple of days are going to help us understand how genetic variation changes throughout just the first few days of development. So on the 4th day after spawning, we started a heat experiment in this conference room. So about half of our animals were held at 26.5 degrees, or that's Celsius, so about 79.7 degrees Fahrenheit. And our heated animals got exposed to 34 degrees Celsius, or 93.2 degrees Fahrenheit. So that's quite a hot day on the Great Barrier Reef. And so in each of these falcon tubes that you're seeing here, there were 50 larvae, and they all stayed under these conditions for a little bit over a day. Once the heat experiments was concluded, all of the animals then got treated to settlement tiles. So this is a little box, probably about yay big, filled with these tiles that we know the animals like to settle on. So about 1,500 larvae went in each container, with the hope that these animals would then start finding places to settle down and metamorphose and start growing as little coral polyps. So we allowed them to do that for a few days. And once that had happened, we started moving all of the tiles back into the main tanks with their parents. And so this is what the animals look like about a month after spawning. So the little white dots on the tiles that you're seeing are the baby corals. So one piece of information that we're now collecting and analyzing, and one of our audience members here today has actually been an amazing asset in helping us to understand, is counting individual coral recruits or polyps on these tiles. So we now have hundreds of photos of these tiles that we took at different time points. And we're now having Ariana here, my amazing intern, count and measure how many baby corals there were in each treatment. So this is going to help us start to understand how exposure to higher temperatures changes the ability of the larvae to actually settle down and start growing on their tiles. Another really cool collaboration that we have coming out of this spawning is looking at the metabolomics and the microbiome of our corals. So for each day before, during, and after spawning, my collaborator Shale was actually taking water samples from our tanks. And so now we're going to process these water samples and try to understand the chemical signatures that the corals may have been releasing, hopefully before, but definitely during spawning. And if we can start to identify the chemical cues that corals are using to communicate with each other, that it's time to spawn, then hopefully at some point we might be able to use some of those chemical signatures to induce spawning later. Another really cool collaboration we have going on is with Cassandra Duran at NASA Ames, just a little bit south of here. And so what we did with Cassandra is I gave her 2 different larvae, so one that had been exposed to higher temperatures and one that had been exposed just to the regular temperature. and 2 different recruits. So again, the recruits are the ones that are just beginning to settle on their tiles. And again, one of them had been exposed to high temperature, one had just been at normal temperatures. And what Cassandra is able to do is actually dissolve these animals into single cells and then sequence each cell individually. And the information that we can then gather from that is how the gene expression in the cells of the heated animals is different from the animals that were at normal temperatures. So that might tell us something about which genes are actually being stressed or affected by higher temperatures. But the other thing that we can do is compare the gene expression between the larvae and the recruits to start to understand which genes are most important in metamorphosis. Another really fun collaboration that we were able to do coming out of this system in December is start to track larval movement. So Manu Prakash at Stanford, his lab has gotten a lot of notoriety because they have this amazing machine called the gravity machine. And what they can do with the gravity machine is track larval movement in 3 dimensions. So this isn't just a normal microscope where you can only see animals moving side to side. What we're actually looking at right now is a coral swimming downwards, which of course is what you would actually see in the ocean, right? These things aren't just swimming in a single plane, they have 3-dimensional motion. And so I've been telling everyone that I think the only person who stared at corals more in December than me, or maybe about the same as me, is Manu's grad student, Ian, who was looking at these animals all the time and took hundreds and hundreds of hours of videos of them so that we can start to understand how these corals decide when and where to move, how they find places to settle, and also start thinking about the biomechanics and the fluid mechanics of how these things actually move in the ocean. So in summary, what are we learning, or what will we learn from the spawning lab? Well, we're starting to get more information about which types of parents are most successful in producing surviving offspring. We're starting to try to figure out how and if the environment selects for different traits in the lab than in the wild. We're also looking into which chemical cues are most involved in spawning, how the microbiome influences spawning, how genetic variation changes throughout just the first few days of larval development, how gene expression changes both during larval metamorphosis and when exposed to higher temperatures, how, where, and why larvae swim and settle, as well as how temperature affects larval settlement. So to summarize, I hope I've convinced you today that coral reefs are both important and threatened, but that lots of research is being done to both document changes in coral communities around the world and to create plans to improve coral health and resilience. And the Coral Spawning Lab here at Cal Academy is aiming to bring some hope for reefs. So at this point, I want to thank everyone who's been involved in this project. I didn't mention, so I'll definitely mention here, our amazing veterinarians, Freeland and Lana, who help to keep these animals healthy here at Cal Academy, as well as everyone else who has made this possible. So I'm happy to take any questions at this time.
First of all, thank you for a highly useful talk. You mentioned that corals spawn with the full moon once a year.
Yes.
So, and you've alluded to chemical signals from them, but how do they know when this occurs? They're sitting in the dark in your basement.
Yeah, it's a great question. And honestly, it's one that people are still working on. So for the question about our basement, our lighting system is set up such that it mimics the moon cycles as well as the day-night cycles in Australia. So we have our lights coming on and off when we want them to, and we actually have a string of blue lights as well that comes on and off to simulate moonlight as well. So that's how we mimic the wild in the lab, but how the corals actually sense that either in the lab or in the wild, we still honestly do not fully understand.
This is extraordinary work. Thank you
thank
so
you.
much. It seems like this is an opportunity to potentially breed for heat-resistant polyps. Are you thinking that direction?
Yes, absolutely. That really is, in fact, my goal, particularly with the heat experiment that we did. My hope is that, and I think it'll take a long time for this to actually be scalable, but my hope is that at some point in the lab, we will be able to show that if you can increase the temperatures when the animals are at the larval stage, the ones that survive those higher temperatures as larvae will grow up to be more heat tolerant adults. And so that's kind of the proof of concept that we're trying to get to at this point.
Thank you very much for this great presentation. Just wondering what the discs, how you came up with the material that is so welcoming to the polyps for settling.
Sure. Yeah. So our settlement tiles, which you can come up and see later if you want, these are aragonite tiles. And to be honest, it was not my decision. It was Rebecca's, who has been working on coral spawning for over 15 years and really knows the ins and outs of what things like to settle on. But I would say there have been a lot of experiments, I think, especially in the last 10 to 20 years, that coral scientists have done to say, what do animals like to settle in and what they don't like to settle on. So aragonite seems to be great. The other thing that we do to make these even more enticing to our little larvae is that we had them sitting in a tank for many, many months before spawning happened. And that tank was actually seeded with, you can actually see it here. You can kind of see it, well, not really in this photo. Let me pull up the first slide. So Crustos coralline algae is this kind of reddish algae that you see in the tank here. And it's actually been shown that coral larvae really like to settle on or near that as well. So we were trying to get as many of our tiles to have some of that Crustos coralline algae as well.
Just one quick last question is, how did you come about picking that particular species that was, not just,
I'm sorry.
Is that the only species that seemed feasible for doing this experiment?
No, I think that, there's lots of different coral species and there's lots of species that spawn. So the two species that we picked for this initial set of spawning kind of pilot projects were the two that have some of the best genomic resources available, because we knew we would want to be doing a lot of genetics with these animals. These are also really animals that are found throughout the Pacific. And so we know that not only are they very ecologically relevant for a lot of Pacific reefs, they're the ones where we knew we would be able to do the most high quality genetics work as well.
We have a couple questions from online, members joining us virtually. So from Meng, what is the plan? Is there a plan to plant them back in the wild?
That's a great question. So for our lab here at Cal Academy, we are not planning to put them back in the wild. Our plan is for us to develop science and plans and tools that inform recommendations for labs to do this much closer to home reefs. So rather than ship animals back across the ocean from San Francisco, what we would like to do is be able to talk to people who want to do this type of work in Palau or in the Caribbean or in Australia or wherever else coral reefs are, so that they can do this in places, on the beach next to the reef and have much easier transplantation that way.
And from another member, how many colonies do you normally keep in a group? Maybe in a tank?
Sure. So in a group, okay. I would say in a tank, we usually want no more than about 5 colonies. Our tanks aren't that large. We have 6 tanks right now. So this is one tank that you're looking at. This has a lot more than 5 colonies because these are the babies, so they're a lot smaller. Again, these are the ones that were born in 2019. So there's about 20 animals in this tank, but I would say for the tanks that have the parents, it's usually 3 to 5.
And Martin asks, how does your work affect any issues of climate change?
Sure. So as we've mentioned before, we did have a heat experiment component for this work in December. And so what we're trying to understand is how we can try to make adaptation to higher temperatures happen more quickly in the lab than it might happen otherwise. Future directions might include trying to expand that work to ocean acidification as well. But at this point, we're really focused on temperature.
Lots of questions coming through. We have another one. Let's see. Did you see any loss of larvae in the tubes at the higher temperatures? And when you released them, it seems like they were released to normal temperature waters. Have you tried releasing to higher temperature waters to settle on the tiles?
Yeah, great questions. So for the first question, really interestingly, in the first 24 hours when those corals were exposed to higher temperatures, we saw slightly higher mortality in the animals that were exposed to higher temperatures, but not a lot. It was a difference of maybe 2 or 3 to 5% difference. But what we saw is a few days later, even once they were back at normal temperatures, the animals that had been exposed to higher temperatures started dying at much higher rates. So it seemed that there was like a little bit of a delayed effect there. And that's actually part of what Ariana is now working on, is seeing how many of the animals that were exposed to higher temperatures actually survived to settle even once they were back at normal temperatures later. So no, at this point, we haven't done that experiment where we keep them at higher temperatures, but we found that even once the temperature goes back to normal, the ones that had been exposed were experiencing more mortality.
See, another question from Meng. Do we know why the vast majority of larvae don't survive in the lab with no predators? Is the survival rate better or worse than in the wild?
That's a great question. So corals are broadcast spawners, which means that they're releasing a huge number of gametes at any given time. There's lots of animals that do this, and the theory behind it is that rather than put all of your eggs in one basket, so to say, or like rather than putting all of your resources into one offspring, you make as many offspring as you possibly can, knowing that very, very few of them are going to survive to adulthood. So even though there are way fewer predators in our lab than in the wild, we still see that phenomenon play out.
And a question from Roger. Will your work contribute to the exhibits for the public at CAS?
Oh, that's a great question. So a few of the baby corals are currently in one of the public exhibits just temporarily. So they are on the Philippine Coral Reef just for a little bit, and then they're going to go back into the lab for a while. My hope is that within the next few years, we could have a full public display of made in San Francisco coral colonies. But yeah, I'll have to do some more persuading.
Let's see, one more question from our virtual audience. From Martin, some fishermen say that their catch is a lot lower than normal. Would raising coral in those areas help this issue out?
Yeah, that's a great question. I mean, certainly all around the world, people are experiencing lower catches in a lot of places. And I would have to know more about which fishery is being affected to know if it's a fishery that would be improved by having higher coral reef cover. But I'm sure that there are places in the world where that is the case. Thank you.
Is the research that you're doing here associated with any particular university?
No, so all of the research that we're doing here is by Cal Academy. I would say we have a number of collaborations. And so, since this event only happens once a year, and since it happens in so few places, I mean, we're really the only lab around that's doing this. We did invite a lot of people from Stanford, from the University of Hawaii, from NASA, from Shedd Aquarium to come and collaborate with us. But I and Rebecca and our other team members are based here at Cal Academy.
And this isn't about corals, but what was the animal that Cassandra Houhan was holding in the picture?
Oh, I think it was a kangaroo. Let me see.
Yeah, I couldn't figure it out.
Yeah, I think it's a kangaroo.
Thank you.
Mhm.
Hi, so you said you were trying to do some breeding for resistances to different temperatures. Were you thinking about doing similar experiments for acidity levels?
Yeah, I think that would be an amazing thing to have happen at some point. I have a friend who once did that for mussel larvae, where they exposed mussel larvae to higher acidity as they were developing, and they saw very different genetic variation in those animals than in the control animals. And so I think that would be an amazing experiment to do in the corals as well.
Hi, I had a question too. So when you were doing the heated experiments, how did you know what temperatures to actually raise it up to?
This is a great question, and one that took a lot of time to try to decide what the temperature should be to bring it up. So we were, first of all, looking a lot at what temperatures are naturally occurring on the reef where these animals came from. So we were looking at the temperature profiles in the Northern Great Barrier Reef, both on a normal day and then on days where they actually observed bleaching. So we wanted it to be hot enough that it would potentially cause bleaching in adults, because that's the reality that we live in, is that more and more animals are bleaching and they need to somehow adapt to be able to survive those higher temperatures. But the other interesting thing that I noticed in the literature is that often the larvae are better at surviving higher temperatures than the adults. And so we actually bumped it up another 2 degrees, thinking that it might take even more to get these larvae to exhibit some type of response, which I think actually ended up being the right call, because we did have a lot of larvae that still survived, even having been at 34 degrees for that long.
I think we have time for one more question.
I think there's one here.
Hi, so certain types of corals spawn. Does that imply that there's other species, genera, or higher that are either almost or exclusively asexual?
Oh, that's a great question. So I wouldn't say that there may be some coral species that are entirely asexual. I can't think of some off the top of my head, but the reason that I've been saying that lots of coral species spawn is that there's another category that are not spawners, but they're actually called brooders. So brooding corals also reproduce both asexually and sexually, but rather than releasing their gametes into the water the way the spawners do, the brooders actually keep their gametes close to home and they brood their larvae within the adult. And so the larvae actually stay close to the parent coral for sometimes up to several weeks. And so that's kind of the distinction that I was making of spawners as opposed to brooders.
Thank you.
Well, thank you so much, Elora. This was a fascinating members talk.
Thank you.
And thank you, members, for joining us in person and virtually. And again, happy Members Month.